So we're pleased to give you a little preview of the 10 research grants that were awarded this year by the CJD Foundation. This is one of the most hopeful things we do each year, and we are so grateful for the support of this program. You can read about all 60 of the grants that have been awarded by CJD Foundation on our website. And you can also view some presentations and papers associated with the grants. So the sources of funding for the CJD Foundation research grants include donations to the foundation, funds raised through Strides for CJD, and memorial grants that are given by a group of families in memory of their loved ones. And in some cases, those grants are built over a period of years. So our grant program has been growing continuously, and since 2015, it has been an annual program. The size of the grants has grown as well, and we now award $50,000 and $100,000 grants. And as a result, the total amount funded on grants has grown continuously, and you can see in the past several years, it's been $500,000 or more per year awarded. If you're interested in funding a memorial research grant, please contact the foundation. You can see the schedule here for receiving funds and awarding scientific grants. So we thank all those who support the CJD Foundation Grants Program. That includes donors, the researchers who apply, and the Scientific Advisory Committee who review the applications. We're going to show you an introduction to all 10 of the grants that were awarded this year. If you have questions about one of them, please feel free to email us, but bear in mind, these are introductions to studies that have recently started, and full reports on each will be given at next year's conference. So now we're pleased to introduce the 10 recipients of the 2022 CJD Foundation Research Grants.
Hello, everybody, and warmest greetings from Hamburg, Germany. I am Hermann Altmeppen, and I am one of the lucky recipients of this year's grant, which actually takes for 2 years, so will end in 23. Although it is a pity that we cannot meet in person, I'm happy to tell you, at least via this way, a little bit about our current project funded by the CJD Foundation. Because of the sad fact that beloved family members or friends were or are affected by this cruel disease, but also thanks to the explanations provided by the CJD Foundation and others, you already know about the basic principles underlying prion diseases, such as CJD. In this scheme, you can see on the left the surface of a neuron and the detrimental misfolding of the normal prion protein, which is here shown in green, into its pathogenic form shown in pink. Our research focuses on enzymatic cleavages occurring in all of our brains right now. An enzyme called ADAM10, which is shown here in orange on the right-hand side, acts like a Pac-Man or molecular scissors and releases the normal prion protein from the cell surface. The project that got funded recently has two connected parts. First, we investigate how this supposedly protective cleavage, a process already provided by nature, can be further stimulated as a therapeutic approach. Previous studies in mice indicated that this is possible. Second, we aim to address whether the released PRP can be measured in body fluids, such as CSF or blood, and serve as a biomarker for earlier and improved diagnosis. As you know, both aspects are critically important to the field and are urgently required. And we have recently generated important research tools, such as highly specific antibodies, that will enable us to fulfill this research. I personally hope, and I think, that from the diverse innovative projects of various researchers funded by the CJD Foundation in the past and in the future, we will eventually end up with a promising combination therapy targeting different aspects of the disease. And therefore, I would like to thank all of you for your valuable donations. In particular, and for this very project, I would like to deeply thank the family members and dear friends of Andy Lewis, who enabled this research, and we will certainly do our best to make it a success. I also want to thank the donors of my previous grants I was lucky to receive via this foundation, since these somehow set the stage for the current one. I strongly hope to meet all of you in person next year. And once again, kind regards from Hamburg, and all the best to all of you and your beloved ones. Thank you.
Good morning, everybody. I am Maria Letizia Barreca. I am a professor of medicinal chemistry at the University of Perugia in Italy. Today, I'm going to provide you a brief overview of our project that this year was founded by CJD Foundation. Just a quick introduction. It is well known that the key event for the development of prion diseases is the conversion of a normal physiological cellular protein called the prion protein, PRPC, into a pathological form that is called PRP-screpy. PRP-screpy then accumulates in the brain of affected patients, leading to neurodegeneration. However, PRP-screpy is not the only cause of these devastating disorders. There are strong evidence that also the normal protein, the PRPC, has some key role in this development of the prion diseases. In particular, PRPC is very important for PRPC propagation, so we can have a larger amount of the pathological form. But what is more important to us is that PRP-screpy needs the normal protein, so needs PRPC to give neurotoxicity. And so in the last years, we have worked in a project with the idea to develop compounds able to suppress, to block the PRPC-mediated toxicity. In particular, about 5 years ago, we have identified a compound that showed some promise in cellular models. Later, we started, to work on this compound to put much efforts, and we have designed and synthesized about 100 molecules. The testing of these molecules led to the identification of some compounds that were very potent in a cellular model of prion diseases. And in addition, these compounds also showed some, I would say, suitable physical-chemical properties for in vivo experiments. And here we are with the CJD Foundation project. So we asked support to test the efficacy of these compounds in an animal model of prion diseases. In reality, we already had in our hands, a mouse model of genetic prion diseases, but we need to validate the model. And this is what we have done in the first part of the project. At the same time, in order to test compounds, you need large amounts. So we have also synthesized the required amounts, and we have the compounds ready. So we already achieved this second task. So you know, at this point, we have the model, we have the compounds, so we are ready for the in vivo experiments. We already asked for some permission because you need some special permission for in vivo experiments. And we are confident that we will start this kind of experiments in autumn. At that point, we will have a proof about the efficacy and also the benefits that our compounds can provide to prion diseases. You can understand very well, I guess, that this is a very complex and multidisciplinary project where we need different expertise and competence. And so I would like to acknowledge my collaborators. At the same time, I really would like all the donors that supported our project, and of course also the CJD Foundation. And thank you all for your attention.
Hi, everyone. My name is Dr. Marcelo Varria. I am currently the head of the Protein Biochemistry Laboratory at the National CJD Research and Surveillance Unit in Edinburgh, United Kingdom. The aim of this capsule is to provide a short overview and some future plans on a research proposal that we recently submitted to the CJD Foundation, and they kindly fund our work. The title of our project is Assessing the Replication Properties of Prion protein aggregates found in archive formalin-fixed paraffin-embedded appendix tissue collected from the asymptomatic population using a highly sensitive in vitro prion amplification assay. As an introduction, I would like to say that variant Creutzfeldt-Jakob disease is a human prion disease that is related to the ingestion of meat products contaminated with a prion disease of cattle. This disease in cattle is referred as a bovine spongiform encephalopathy, or BSE. Despite widespread human exposure to BSE in the United Kingdom, the number of variant CJD cases remain low, with 178 cases currently reported. However, the possibility of human-to-human variant CJD transmission and the uncertainty over the prevalence of asymptomatic carriers instigate 3 large retrospective studies in the United Kingdom. These studies, named as Appendix 1, Appendix 2, and Appendix 3, looked at the presence of the abnormal form of the prion protein in formalin-fixed paraffin-embedded appendix tissue. These studies estimate that 1 in 2,000 people are potential carriers of this abnormal form of the prion protein in the United Kingdom. Also, it's important to mention that a fundamental characteristic of this abnormal form of the prion protein is that it's able to replicate and preserve its anomalous shape, usually found in protein aggregate, at the expenses of the normal form of the prion protein, the PRPC. We mimic this replication process in the test tube using a method called highly sensitive protein misfolding cyclic amplification, or HSPMCA. This technique is able to replicate and detect tiny quantities of the abnormal form of the prion protein in human tissues and biological fluids. The aim of our study is to explore the nature of the abnormal prion protein aggregates found in these anonymized archive appendices to find out if they truly represent any link with human prion diseases, but particularly with variant CJD. For this project, we will be investigating particularly cases homozygous for valine at the codon 129 of the prion protein gene, and they are linked to the Appendix 2 study. We hope to present our result at the next 2023 CJD conference, but before that, I would like to thank the people that contribute and supported our work. So hopefully see you next year. Bye.
My name is Joaquín Castilla. I'm the principal investigator in the Spanish Research Institute CIC-Biobune. The project I'm going to talk today the name is Development and Characterization of the Fastest Animal Model Able to Propagate GSS prions Faithfully. The development of therapies for any disease requires previous assessment of the safety and efficacy of such potential treatments in animals. To assess the potential effectiveness of the treatment, it is important to count with animal models that faithfully reproduce the disease observed in humans. The chances of potential therapies to fail in human trials depends, in many cases, on how similar both are. For that reason, through this project, we intend to develop and characterize a new animal model that is directly susceptible to human prions, particularly to prions causing Gerstmann-Straussler-Scheiker disease. Apart from allowing testing the effect of therapies on human prions before facing clinical trials, this new model offers other important advantages. According to our preliminary results, this might be the fastest animal model developed for a human prion disease, allowing to accelerate the trial of potential treatments and therefore reducing the cost of treatment development. Additionally, biomarkers indicating disease progression, molecules that preclude the onset of the neurological symptoms, have been detected in the blood of this model, animals in this model. All these characteristics would make this animal model ideal to test rapidly potential therapies for prion diseases, although the model requires further characterization to make it feasible. Throughout this project, we will evaluate whether other human prion strains could be even faster. We will test different inoculation routes in order to make it usable for treatment targeting prion transport to the brain. We will fully characterize the lesion appearing in the brains of these animals to evaluate disease-modifying therapies. And we will demonstrate the utility, assessing the efficacy of two potential treatments. One aimed to reduce PRP-C, cellular PRP levels, and another one using a molecule with anti-prion activity in vitro. I want to thank to the supporters that financed this grant that I'm pretty sure is going to be very useful for the whole community, prion community. Thank you very much.
Hi, my name is Leonardo Cortes. I'm a research associate in Dr. Valerie Sim's lab at the Center for Prion and Protein Folding Diseases in University of Alberta. So first of all, I want to take this opportunity to thanks very sincerely to the CJD Foundation and especially to the donors that decided to support our research. Today, I want to give you a brief overview of this project that focused on studying how different types of prions drive different disease phenotypes. We know that prion diseases can be presented with a broad and variable range of symptoms. We also know that during prion disease, the normal prion protein misfold, change its shape, and start clamping together to form a wide variety of prion particles. Prion with different shapes and different properties. We can divide these prion particles in two groups, two populations. One that include the smallest particles, the particles that are less stable and possibly more toxic. And a second population that include the larger particles, more stable, much harder to be degraded by the brain. These two populations of prion particles also differ in the ability to replicate and spread in the brain. So in this context, a natural question that comes to our minds is there any association between these two populations of prion particles and the different symptoms that a patient with CJD present? Well, in an article that we recently published, also with the support of the CJD Foundation, we found that in fact there is an association. We proposed that the different proportion of these two populations of prion particles in the brain of patients with CJD will drive their symptoms. However, there is a very relevant aspect that we haven't explored yet, an aspect that will help us to know more deeply about this association between type of prion particles and symptoms. And that aspect is the regionality. With regionality, what I mean is figuring out how these two populations of prion particles spread and how they grow in different regions of the brain. So each brain region is a specific, a very unique environment that can favor one particular population or one particular type of prion. Then the consequence of that is that we will have different degrees of damage in different regions of the brain, with the consequent different symptoms for that disease. For example, if more neurotoxic prion particles accumulate in the occipital region of the brain, that patient will have vision problems. So in this project, using the same techniques and same method that we used before, we will try to isolate and characterize the different type of prions accumulated in different parts of the brain of patients with CJD. This will allow us to understand better why different patients have different symptoms. And ultimately, this knowledge could help us to better target specific type of prion based on the symptoms of the patient. Another interesting question that we will try to answer is which factors control the propagation of these two populations in the brain of a patient with CJD. We know that the prions are formed in the membrane of the neurons, and the membrane is a particular structure that is composed mostly for lipids, a specific type of molecule that is present in all these membranes. So what we will try to do is try to identify which lipids are present in each region of the brain and try to associate the type of lipid in that region with the type of prion that is accumulated in that specific region. If we identify which lipid influence the propagation of specific type of prion, then we will have a new target toward which to direct our therapeutic strategies. So those are our goals, and with your support, we will try our best to get there. So thank you very much again. Thank you especially to the donors. And hopefully next time I will be able to show you exciting results.
Hello, everyone. My name is Catherine Hague. I am coming to you today from the Rocky Mountain Labs in Montana. I'm very excited to be telling you about some of the work that we're doing. Our grant was called Regional Tropism of Sporadic CJD Subtypes in Human Brain Organoids. So what we really want to look at here is that we know during sporadic CJD, patients will present with different symptoms and also different disease durations. And they are often caused by the different subtypes, which we think are just really very small changes in the structure of the prions, the agent that causes disease. But those very small changes seem to allow this agent to attack different parts of the brain. And by attacking different parts of the brain, you get the different symptomology. So we want to really delve into how these different shapes attack the different parts of the brain, and maybe even different cells within those different parts of the brain. Until recently, that hasn't really been possible because we need a model of live human brain tissue to do this. But a few years back in time, my lab developed this. And these are human cerebral organoids, or human brain organoids. They're balls of brain tissue we grow in a dish. They can grow to about the size of a small pea. We actually grow them from stem cells that we reprogram from people's skin. So it's very ethically appropriate. All of our donors have consented to us using their skin to make these little brain cultures. And we were able to show that we could infect them with sporadic CJD prions. So what you see here, this brown, is accumulation of the disease-associated prions within the organoids. Since we did this, organoid technology has actually improved immensely. And now we don't just make these organoids left, right, and center. We can make them correspond to different parts of the brain, which allows us to look at those different subtypes, those different shapes, and how they attack the different regions. And that is what we propose to do here. We're going to make these different parts of the brain organoids, and we're going to infect them with our different subtypes, our different shapes, and look at how the cells within those organoids are affected. The way we're going to look at the cells is to use a new technique, or fairly new technique, called single cell sequencing. What this does is it looks at the genes that are expressed within the organoids. So you are defined by your genes. It defines your hair color, your eye color. It's the same with the cells within the organoids. Their genes define them, which means we can identify them. And the readout we get looks a lot like this. Each dot is an individual cell. And this is kind of the family tree of cells. That the cells that are most closely related, i.e. the same type, group together, and the distant relatives are, say, over here. Once we've got this data, this allows us to look in detail at what's going on in this type of cell versus these types. And we can see how the different subtypes are affecting these populations differently, or the same, or maybe not at all. And what we really hope that we will be able to pull out of this is the weaknesses that allow a subtype to say hit these cells and not these cells, or both. And if we can define what those weaknesses are, ultimately, we really want to then try and intervene to prevent that weakness being targeted, stop the disease right at the very beginning, or find another way to intervene to stop those cells being damaged and eventually dying during the disease. And so I would like to thank all of you, the donors. This work is incredibly complex, time-consuming, and very, very expensive. And we honestly could not do it without your help. This grant really makes the difference between us being able to do it and not. So thank you ever so much. We really appreciate you.
So my name is Rodrigo Morales. From the University of Texas Health Science Center at Houston. And the title of the project that we are planning to pursue is the development of a high throughput system for screening antiprion molecules. So why we are studying this? So as you know, prion diseases affect humans and several animal species, but prion diseases is not one disease. As you know, in humans, for example, we have different types of Creutzfeldt-Jakob disease. We have Kuru, we have a fatal familial insomnia. So all these diseases are thought to be caused by different strains of infectious prions. So what are these strains of prions? So we believe that there are different conformations of the infectious agent, different shapes of the infectious agent. So if we think on these different strains as different conformations of prions, it is easy to think that a certain drug attacking one conformation will not be doing it in the same way for a second strain, having a different shape. So that's why we believe that, for example, some previously identified antiprion molecules has been again identified in rodents, have not been active in human prion diseases. So what we are trying to do in this project basically is to develop a system that will allow us to screen libraries of small molecules with the hope of identifying strain-specific antiprion molecules. So For that, we are taking advantage of the PMCA technology, the protein misfolding cyclic amplification technique, that is able to replicate prions in a very fast manner. And fortunately, this technique also is able to maintain the conformation of the infectious prions that it is replicating. So in that sense, we can test whether a specific compound is able to inhibit the propagation of a specific prion strain. So in this project, what we are planning to do is to work in two different aims. In the first one is to standardize this high-throughput PMCA platform that we call for two different animal species. We are planning to do it for human prion strains, but also for deer prions. So of course, the relevant thing here is to do it for human prions, but it's critical also to do it for deer for two reasons. One is because of the problem that chronic wasting disease prions are making in North America, especially in the United States, but also is because we want to test whether molecules identified as active for human prions are also able to be active for deer, or they are exclusive for humans. And the other way around, we want to see whether some molecules active against deer prions are able to go against the human prions or not. So we will develop this in two different formats. Hopefully we will go for a 384 well-plate format that will allow us to test a higher quantity of molecules. And once this is standardized, what we are planning to do is to screen these large libraries of molecules in this high-throughput PMCA technique, with the hope, again, of identifying strain-specific antiprion compounds. So what we have done so far, we have standardized the high-throughput PMCA technology for 6 different rodent prion strains, and we have identified some strain-specific antiprion molecules. So we know that this principle works using specific and well-established prion strains generated in the laboratory. So considering that, we want to jump now to the next step, testing the same principles for more relevant human prions and deer prions. So with that, I would like to conclude. I would like to thank the different awards that are making possible this research. And I really hope to bring good data and good results for the next year meeting. Thank you very much.
Hello, everyone. My name is Jamie Noguez. I am the Section Director for CSF Testing at the National Prion Disease Pathology Surveillance Center at Case Western Reserve University in Cleveland, Ohio. So this is the only center of its kind, and it offers diagnostic testing for prion disease to patients and their families. So the title of my research study is Improving the Sensitivity of CSF RT-QuIC in Humans by Examining Cases with Indeterminate Results. And in the next slide, I'm going to tell you a bit about it. So one of the tests that we offer at the Prion Center is called Real-Time Quaking-Induced Conversion, which we abbreviate as RT-QuIC. Now, this is a very good test for detecting prion disease using cerebral spinal fluid samples from patients to look for the misfolded protein. It's a test that has about 90% sensitivity and about 95% specificity. So what that means is that it's very good at detecting prion diseases, but it's also good at differentiating it from other types of neurodegenerative conditions. But we know that there's still room for improvement. So about 10% of prion diseases are known to give false negative CSF RT-QuIC results. And we know that about 1% of CSF samples that we see come through the Prion Center, they yield an indeterminate CSF RT-QuIC result. The image on the right shows examples in columns A and B of characteristic curves for positive and negative results. Now, these are very straightforward to interpret. But then we get samples that have results like shown in columns C and D. So these are indeterminate results. And as you can see, these don't look quite positive, they don't look quite negative, and so that presents a challenge at times for us to interpret them. So our hypothesis is that by further examining indeterminate cases like these, we may be able to find ways to improve the sensitivity of our current CSF RT-QuIC tests. And by doing that, we'd be able to improve the diagnosis of human prion disease, especially in these rare cases or in the subtypes that we know most often result in false negative CSF RT-QuIC results. So how are we going to go about doing this? So my study involves 3 aims, and they're as follows. First, my goal is to determine the final diagnosis of patients with indeterminate CSF RT-QuIC testing results, and I plan to do this by reviewing their autopsy records. My second aim is to determine if there are any characteristics that these patients that were diagnosed with prion disease and had indeterminate test results, are there any characteristics that they have in common, such as disease subtype, age, sex, etc. And then finally, I plan to see if there are any ways that we can modify or optimize the current CSF testing protocols in order to improve the CSF RT-QuIC test performance in these cases that have indeterminate and false negative results. And these are going to be for patients with a final diagnosis of prion disease, but then have these indeterminate and negative results. And so we have an archive of these specimens at the Prion Center that I'll be using to help me do this. So with that, I'd like to very sincerely thank the donors listed on this slide for the grant that's funding my research. I truly appreciate the opportunity to conduct research that I know is going to make a difference in the lives of patients and their families. So please accept my deepest gratitude. Thank you.
Hello, I'm Shashi Shetty. I'm a researcher at the National Prion Disease and Pathology Surveillance Center at Case Western Reserve University. And I'm also affiliated with the University Hospitals Cleveland Medical Center in Cleveland, Ohio. My research title is Examining Copy Number Variants as a Possible Phenotype Modifier in Prion Disease. I'd like to state the 3 goals that I'm focusing on for this research. One is to examine the role of copy number variants in young age at onset, sporadic prion disease, such as sporadic CJD, the VV1 subtype, and sporadic fatal insomnia. My second goal is to identify any novel CNVs in genetic prion disease so that which can result in discovering new genetic risk factors. I'll get into a little bit of the details. And then presence of CNVs in the brain. So when you find these CNVs, does this really affect the function, or does it cause the disease, or does it increase susceptibility? We will be doing some functional studies. My group, that is Dr. Appleby, Dr. Callie, and myself, the 3 of us are going to focus on looking into the comprehensive nature of CNV, not just finding it, but also what does it do functionally. So that is the goal of our research. So to give you a little bit of background, copy number variants, these are small stretches of DNA in our genome that can be deleted, duplicated, or rearranged in the genome. And this is important. It happens in normal population. Like if we tested ourselves, we will find those variations in our genome. It is important for human evolution, and it happens in other species as well. So it is important. The copy number variation is very important for our evolution. However, if this copy number variation happens rarely in a region that contains genes that could impact health or disease, that's the one we are trying to target. We are trying to see if there are rare variants or rare copy number variants that could be predisposing either to prion disease or modifying susceptibility or age of onset. So keeping that in mind, one of the things we want to target is the sporadic prion disease. By sporadic, I mean, we don't know why prion disease happens. We know that it is because of the prion protein, misfolding of the protein, but what is that is predisposing a person? Because this is such a rare disease, 1 in a million. So why does it happen in one person, not the rest of the population? So we are trying to understand what is going on in the genome. Could these copy number variants be predisposing to the disease, either early age of onset, or it could be making the phenotype more severe or mild? We'll start with sporadic prion disease, especially the VV1 subtype and of sporadic fatal insomnia, and look for in these young patients, typically prion disease happens in older individuals, like in their 60s. But in the VV1 subtype and SFI, it is seen in very young people, young individuals, like in their 20s, which doesn't make sense. Why is it happening in these young people? Like, when it is supposed to be happening, it's so rare, happening in 1 in a million, and in people who are older than 60. And here it's happening in somebody who's 18 or 20. What is predisposing? What is the factor, predisposing factor? And then the second type is the genetic type, where we know that the PRNP gene is a causative gene. So any mutation or defect in that gene is causing genetic prion disease. And these are typically inherited from a carrier parent. So somebody who's carrying the mutation, who at some point may develop the disease. So again, with the same mutation within family, say there are 4 people who have this mutation in the family, but you'll see that someone may never develop the disease. Another family member might develop it at a young age, or someone else may develop it later on. So there is variability, like intra-family variability. Within the family, we see the same gene, same mutation, but the effect is different. Either they never develop the disease, or if they develop it, it's very young, at a very young age, or they might develop it at a later age. And the phenotype, like the expression, the cognitive decline, it might be slow, could be fast. So there are variability, and it doesn't make sense that this prion gene is behaving differently. The same mutation is behaving differently. So we are trying to figure out if these CNVs, the rare novel CNVs, could they be the risk factors? Either they are being protective, just presence of these stretches of DNA that are variable. Could that be protecting a patient or an individual versus not protecting or making the other person more susceptible? So once we identify these CNVs, we want to know, are these CNVs did we just happen to see it in the brain? Is it truly causing the disease, or is it making the person susceptible, or is it showing like a protective effect? So how do you do that? We want to do functional studies for that. I look forward to sharing my research work in the near future, and I would like to thank the donors for supporting this research. Thank you.
Hello, everyone. First of all, I would like to thank the CJD Foundation for this opportunity for me to update our research progress. I'm Wenquan Zhou, professor from the Department of Pathology and the Associate Director of the National Prion Disease Surveillance Center at Case Western Reserve University, Cleveland, Ohio. I'm the principal investigator, along with Dr. Kitsumi Duhuna, and also we have 2 key co-investigators, Dr. Qinzhong Kong from the Case Western Reserve University, and Dr. Ruth Gabezon from Hadassah University Hospital, Israel. The title of our grant is Assessing Prophylactic and Therapeutic Efficacy of Cellulose Ether CE Compound TC5W on CJD. CE compound, including TC5W, have been widely used as additives in food, pharmaceutical tablets, and personal care products. Duhuna and the co-workers first discovered that the CEs are able to inhibit animal prions and increase survival rates of prion-infected animals. Recently, my lab has revealed that the TC5W is also able to inhibit the human prion formation and directly reduce human prions in cell-free models. Our new study, supported by the CJD Foundation grant, is to further examine the inhibitory effect of this compound on human prion diseases with 2 lines of humanized transgenic mice. The first line is TGFBI mice that express human prion protein from Dr. Qingdong Kong's lab to examine its prophylactic effect. We give the animals the CE compound subcutaneously, followed by infecting the animals with human prions to determine whether the compound will prevent the prion disease development. To examine its therapeutic effect, we first infected the animals with the human prions, and then after approximately 5 months, the animals will be given the compound to determine whether this compound can cure prion disease or not. The second line is TGE199K mice expressing human mouse chimeric mutant PRP E199K that mimics human genetic CJD with the PRP E200K mutation, the most common mutation in Caucasian population. To examine its prophylactic effect, the mice were given the CE compound subcutaneously at the very young age of 2 months to determine whether the compound will prevent the mice from developing the prion disease. To investigate its therapeutic effect, the mice were given the compound at approximately 230 days, when the mice already started to show the prion-related signs. Our preliminary observation reveals that the mice treated with the compound are doing better than the mice without the treatment, according to the well-being score, which is highly encouraging. Finally, and most importantly, we would like to thank all donors for their generous support to our study. Thank you for your attention.
Thank you for joining us for the final session of the 2022 CJD Foundation Virtual Conference. We were glad to share this preview of the studies recently started by the recipients of this year's research grants. They will each provide a full report at next year's conference. In the meantime, if you have a question about today's session, please contact us at the CJD Foundation. Thank you for supporting the CJD Foundation and our research grant program. Have a good evening.